Let's talk about the autonomic nervous system, or the ANS. So the term autonomic is derived from the root auto, that means self, and nomos, meaning law. And the ANS, it regulates the functions of involuntary organs. Now this is crucial right here. Involuntary organs, which include the heart, the blood vessels, the exocrine glands, and the visceral organs. So you don't have to pay attention and tell your heart to beat. It will beat automatically. You don't need to tell your blood vessels, hey, I need you to vasodilate a little bit, or I need you to constrict. They do it on themselves. You don't need to tell your lacrimal glands to start secreting fluid in your eyes so you can start crying or you can get something out of your eye. It does it automatically. And the gut, it will digest food automatically. So this is the autonomic nervous system. The ANS is usually subdivided into three divisions. Classically, the sympathetic and parasympathetic, and then the enteric division of the autonomic nervous system isn't talked about much, but mostly we'll just focus on the sympathetic and the parasympathetic parts of the autonomic nervous system. So something crucial here to remember is the sympathetic nervous system, or part of the autonomic nervous system, is adrenergic receptors for the adrenal-like actions resulting from the sympathetic nerve activation. So when you hear of an adrenergic receptor, think sympathetic nervous system. And the reason why is because adrenaline, is also known as epinephrine, acts on these adrenergic receptors. So think sympathetic nervous system. When you think of the sympathetic nervous system, think adrenergic receptors. The parasympathetic system was designated as cholinergic for acetylcholine, acetylcholine-like action. So the acetylcholine is the neurotransmitter that acts on cholinergic receptors, and think cholinergic, parasympathetic. So adrenergic, think sympathetic; cholinergic, think parasympathetic. So the sympathetic division is said to preside over the use of metabolic resources and emergency responses. So the classic example is fight or flight, right? The sympathetic division is over the metabolic resources when you're undergoing a lot of metabolic activity and under emergency responses. The classic example is a guy being chased by a tiger. You got to fight or flight, survival of the fittest. But no one nowadays is chased by tigers, usually. But it's more of like if you get scared, or if you driving down the road and then you're going way too fast, and then the cop flips on his lights and starts coming up behind you, and you feel that wave of fear and emotions, anxiety, all that going over you. That is the sympathetic response. The parasympathetic division presides over the restoration of the body's reserves and the elimination of waste products. In reality, most of the organs supplied by the ANS receive both sympathetic and parasympathetic innervation. And so this is going to be crucial, is because most organs, so if I have an organ right here and it has symp coming into it, it usually has parasymp coming into it too. So this is an organ. So if I take out the sympathetic part of the organ, then there's going to be more parasympathetic activity. If I take out the parasympathetic activity, then there will be more sympathetic innervation into the organ. So that will either slow it down if the parasympathetic trumps the sympathetic stimuli, then the organ will usually be shut down. And this depends on the organ. If it's a visceral organ or some kind of gut organ, it will increase its function. So let's say I have a heart here. And I have sympathetic coming in, and then this side I have parasympathetic. So if I cut somehow all the sympathetic nerve innervation to the heart's going to slow down, because the parasympathetic system usually is what? It reserves energy and it eliminates waste. But so in the case of the heart, if I cut all the sympathetic fibers to the heart, the parasympathetic is still innervated, so the heartbeat will slow down. If I do vice versa, where I cut only the parasympathetic innervation to the heart, well, then the sympathetic nervous system is still going to be able to really cause heart rate and all that stuff to increase. So organs that have both sympathetic and parasympathetic innervation, they can act in that manner. Now let's say I have another organ and it only has sympathetic innervation, because parasympathetic innervation, the nervous system doesn't send parasympathetic fibers to every part of your body. It does send sympathetic innervation to every part of your body. All your blood vessels have to have sympathetic innervation. All parts of your body have to have sympathetic innervation. So let's say it just has sympathetic innervation on this organ, and the parasympathetic, and I'm going to explain the anatomy and all that behind this, but I just want to get this point across, is that if I take out the sympathetic innervation to an organ that just has sympathetic innervation, then it might act as if parasympathetic innervation is involved. So if I get if I get here neurotransmitters coming into this organ, and then I take it out by blocking a receptor or something, then this organ will no longer, because this is like the gas pedal in a car. If I take my foot off the gas driving a car, it will slow down, but not at the same rate that if I apply the brake. And the brake could be the parasympathetic. So if an organ just has sympathetic innervation, if I take it off, it's analogous just to taking my foot off the gas. The organ will slow down, or it will decrease its function, or whatever this sympathetic innervation is. If I take it out, then it will do opposite of that, but not at the same rate as if I applied parasympathetic innervation to the organ and then had this going full bore. If that makes sense, I hope it does. Okay, so the autonomic nervous system consists of a 2-neuron efferent chain. So this is important. So this is kind of a autonomic nervous system versus a somatic motor neuron. Okay, so here is the anterior horn of the spinal cord, and at every vertebra, you got a spinal nerve coming out. And in a somatic motor neuron, you have the anterior horn located. This cell kind of starts right here in the spinal cord, and there's one continuous cell that comes right out to this, what they call the neuromuscular junction. This is your skeletal muscle system. This is voluntary. You do have control over your muscles, over your skeletal muscle system. So this is the somatomotor system. And acetylcholine is the neurotransmitter here for the neuromuscular junction. And we'll see acetylcholine in the parasympathetic nervous system too. However, in the autonomic nervous system, both parasympathetic and sympathetic parts of the autonomic nervous system, you have a two neuron. And so they have the preganglionic and the postganglionic. All right. And then the preganglionic nerve, the cell bodies start right here in the intermediolateral horn. They can start elsewhere, but in this example, it's the intermediolateral horn cell. The fiber goes out and it synapses onto a postganglionic axon. Okay? And right here where all these synapses take place, because this is a cell body here of the postganglionic cell, there's ganglions, and we're going to talk about the anatomy and where these ganglions are located. But basically, you have two neurons. You have two neurons, the preganglionic and the postganglionic. And something's interesting that I didn't realize until recently is that the postganglionic axons terminate in varicosities. And what are varicosities? They're swellings that contain synaptic vesicles, which release the neurotransmitter into the extracellular space surrounding the effector cells. So these are effector cells. This could be the adrenal glands. This could be the pupils. This could any autonomic innervation. These are the cells. And so there's little varicosities. I always thought it was something like this, where the end of the terminal just kind of synapses on every part of your body, but that would be incredibly taxing to have one cell come out and innervate each, or one neuron come out and innervate each cell. This would be ridiculous. So there's varicosities. So let's say all of these are heart cells, and then the nerve kind of comes through this part. Well, there's these varicosities here, and these varicosities are just little swellings with synaptic vesicles. And when it gets stimulated, this postganglionic nerve gets stimulated, then all these varicosities, they release their vesicles, and they diffuse out into the cells. And so that is pretty cool. So let's talk about first one neurotransmitter, acetylcholine. And so in the autonomic nervous system, acetylcholine is released in the following sites: all pre and postganglionic parasympathetic neurons. Okay, so that acetylcholine works on the all pre and post parasympathetic ganglions. Do you remember what kind of receptor are in the parasympathetic neurons? Cholinergic. Okay, acetylcholine, cholinergic. You're going to have to remember that. So this is an image of the synapse. And so here you have glucose that we eat in abundance. A glucose gets converted to pyruvate and then gets shipped to the mitochondria. And then part of the Krebs cycle, or glycolysis, you get acetyl-CoA. Okay, so acetyl-CoA gets paired up with choline, and there's a sodium choline transporter at the end of the neuron. And so choline, because the sodium potassium pump is constantly pumping out sodium here, we have an abundance of sodium outside, or extracellular, outside the cell. And so there's a transporter there that brings in one sodium and one choline. So the choline comes in. That's how choline gets inside the nerve. Sorry, the neuron. And so they get paired up by choline acetyltransferase, and that's how acetylcholine gets made. The acetylcholine then gets shipped into vesicles, and you have the vesicular acetylcholine transporter. And then once the nerve is stimulated, acetylcholine gets shipped into, or gets excreted into, the synaptic gap. This is called a synaptic gap, or synapse. Synapse, or synaptic gap, synaptic cleft. There's a lot of different names for it, but this is the synapse. And so once acetylcholine is inside this synapse, then the enzyme acetylcholinesterase begins to break down acetylcholine into acetate and choline. Choline then gets shipped back, gets recycled. And so acetylcholine, why it's in this synaptic gap, it stimulates the effector cells, or the postganglionic cell, to do its function. Whatever this cell is going to do, designed to do, that's what it will do when acetylcholine gets onto these receptors. That's acetylcholine. Also, acetylcholine is used in all preganglionic sympathetic neurons. So here we have acetylcholine is the neurotransmitter so this is the parasympathetic division, okay? And this is the autonomic nervous system. Here's the parasympathetic here's the parasympathetic branch, and here's the sympathetic branch. Remember that acetylcholine is in all pre and post parasympathetic divisions. So then you have acetylcholine, which is right here in this synapse, and then you have acetylcholine, which is used in this synapse. And they're cholinergic receptors. And there's two types of cholinergic receptors. There is nicotinic and there is muscarinic. Okay? And the nicotinic receptors are, if I go back here, these are nicotinic receptors. Acetylcholine receptors, these are nicotinic. So these are the nicotinic receptors right here. So if you're talking about either the preganglionic cell of the parasympathetic or sympathetic division of the autonomic nervous system, the first synapse outside of, because there's synapses all through this CNS, you know, but once you leave the CNS, the first preganglionic cell or neuron synapses on a nicotinic receptor, and acetylcholine is the neurotransmitter. And so the nicotinic receptor was named because of nicotine acts on the nicotinic receptor, and so they called it the nicotinic receptor. And that's just what it was named. This is also here on the parasympathetic division, the postganglionic nerve. There's a receptor called muscarinic receptor. And why is it called the muscarinic receptor? Because muscarine is a natural product found in certain mushrooms. And so muscarine was isolated in 1869, and it was the first parasympathomimetic substance ever studied and causes profound activation of the parasympathetic nervous system. So they called these receptors they found at the postganglionic synapse of the parasympathetic division the muscarinic receptor. And so another word right here is the parasympathomimetic, is that mimics the parasympathetic nervous system. So all drugs that are parasympathomimetic are drugs that mimic this muscarine or this acetylcholine or any kind of neurotransmitter here that works on these muscarinic receptors. And here's the chemical of muscarine. So nicotinic receptors are in the first synapse of the parasympathetic and sympathetic divisions of the autonomic nervous system, the preganglionic to postganglionic synapse, and the muscarinic receptor is on the postganglionic parasympathetic division, and acetylcholine is the neurotransmitter of all three of these locations, 1, 2, and 3. Now here you have this adrenergic. This is adrenergic. I don't know if I spelled that right, but adrenergic receptor is here. And you have alpha or beta adrenergic receptors. Adrenergic because of adrenaline acts on these types of receptors. So the muscarinic receptor is of the indirect ligand-gated type, uses a Gprotein. So the muscarinic is a Gprotein. And the nicotinic is a direct ligand-gated type. And this makes sense because the nicotinic receptor, you know, you don't need to take time to get this across. And so this direct ligand-gated is just a ligand, and it means it's an ion channel. And so these ions are going to just rush right through, transfer the signal right on down the line. But sometimes these Gproteins, they take a while to get going, and because they have to turn the second messenger system on, and we'll talk about that. but the muscarinic, the parasympathetic division, is usually a Gprotein. It takes a little bit longer than these ion channels in these synapses here. So the catecholamines. What are the catecholamines? You have epinephrine, which is also called adrenaline. You have noradrenaline or norepinephrine, and you have dopamine. So how are these catecholamines produced? First, you need tyrosine. Tyrosine is then converted to dopa, which is then converted to dopamine, which is then converted to norepinephrine, and which is finally converted to epinephrine. And here are the enzymes that cause that transformation. So this picture is of a adrenergic or a sympathetic post-sympathetic ganglion synapse. And so they have adrenoreceptors or adrenergic receptors. And so let's kind of walk through this process. So first of all, tyrosine needs to become, remember that tyrosine is the precursor to norepinephrine and epinephrine. And so tyrosine needs to come into the cell. So the same as in choline, you need a membrane protein here that will transport sodium and tyrosine. It's a kind of a co-transporter here. And remember that sodium is higher concentration on the outside than in the inside because of sodium-potassium pumps. And so the sodium and tyrosine are going to be shipped into the end of the postganglionic nerve at the same time. So then tyrosine through tyrosine hydroxylase, right here, this enzyme right here, is going to convert tyrosine into dopa. Now we come in contact with a drug. This drug right here, mettyrosine, blocks this enzyme. So norepinephrine ultimately will not be secreted into the synapse because mettyrosine blocks it. Dopa then is converted into dopamine by this enzyme right here. Dopamine then is transported into a vesicle, and hydrogen is exchanged out. And we come into another drug, reserpine. Reserpine blocks this VMAT protein here, and so norepinephrine or dopamine cannot get inside here to become norepinephrine. So once norepinephrine is kind of inside this vesicle, and this is just one, but there's hundreds, thousands of these vesicles all over the end of the synapse. Calcium, because of an electrical signal coming down the neuron, this calcium channel opens up. This calcium then comes in, binds to these VAMP proteins, and these VAMP proteins then open up and fuse, and then fuse to the membrane here, and you get norepinephrine released into this synapse. This is a synapse here. Synapse. And SNAPs proteins and VAMP proteins, they're all related, and you can study those, Google those. But here we have another couple drugs, bradylilium and guanethidine. These block these receptors so that norepinephrine will not be released into the synapse. So once norepinephrine is into the synapse, it crosses the synaptic cleft, binds to these adrenoreceptors, and produces an effect after binding. So then the norepinephrine gets diffused away from the synaptic cleft, and this norepinephrine transporter, this NET protein, reuptakes, kind of takes norepinephrine out of the synapse and puts it back up into the cell so it can be recycled. Cocaine and tricyclic antidepressants block this receptor so that more for a longer period of time, norepinephrine is in this synaptic gap, affecting these receptors, stimulating them to produce whatever effect they're going to produce. Also, there's an autoreceptor here, so norepinephrine comes back and hits this, and I think that this comes in and this blocks this too. Well, oh, sorry, it blocks this. It blocks this pathway because once norepinephrine comes up and stimulates this autoreceptor, well, then the cell's like, hey, we already got enough norepinephrine in the synapse. You don't need to release any more. So that's a negative feedback. So norepinephrine and epinephrine, or adrenaline, they affect adrenergic receptors. Where are adrenergic receptors located? They're located on the sympathetic postganglionic synapse. Adrenergic receptors are classified as either alpha or beta. So when you get alpha blockers or beta blockers, these are the adrenergic receptors that are being affected. So they're subclassified into alpha 1, alpha 2, and beta 1, 2, and 3. The adrenergic receptors are the indirect ligand-gated Gprotein link type. So the adrenergic receptors are Gprotein receptors. And the alpha 1 receptors, they activate phospholipase C, and they increase the intracellular concentrations of diacylglycerol and inositol triphosphate. We have pictures coming up about these. And the beta receptors type stimulate adenylyl cyclase, whereas the alpha 2, they inhibit adenylyl cyclase. So let's talk about the Gprotein and phospholipase C pathway. So you have a stimulatory signal, in this case norepinephrine or epinephrine, and dopamine. They'll come in here, they'll activate this receptor. This is called a Gprotein coupled receptor, GPCR. So you'll see this in the literature. This is a Gprotein coupled receptor. What this means is this is just the receptor, R, but it's coupled to a Gprotein. So the Gprotein is on the inside surface of the cell. This is the outside, this is the inside. The Gprotein, once this is activated, the Gprotein comes and gets coupled with this receptor, and then this has GDP bound to it. The GDP is kicked off, and the GTP is kicked on. Now there's alpha, beta, and gamma subunits to this Gprotein. Well, there's different types of Gproteins, but the most common is, or that's talked about, is there's 3 parts to it, the triheterodimeric, or I can't remember exactly what they call it, but there's an alpha, beta, gamma subunits. So the alpha subunit, once it has that Gprotein to GTP bound to it, the alpha subunit comes over here and it activates PLC, which is phospholipase C. Phospholipase C takes PIP2, which is part of the cell membrane, and converts it into IP3 and DAG. So IP3 can be converted to IP4, and then that causes calcium entry across the plasma membrane. The IP3 can be broken down into IP2, which is used for kind of recycled to go back up into form the PIP2 again. And so the IP3, what happens is it causes the endoplasmic reticulum to secrete calcium or to release calcium. So once this DAG, and we'll stop right here and catch up on this side of the phospholipase C pathway, the DAG then comes over and turns on protein kinase C, or PKC, protein kinase C. And what protein kinase C does is it activates certain molecules inside the cell, and you have a cellular response. Okay, but so once this calcium through IP3, IP3 then attaches to the endoplasmic reticulum, and then calcium comes out into the cell. The calcium also activates protein kinase C, as does the DAG, and it also causes this CAM kinase to be activated. And so once this CAM kinase becomes activated, it also causes cellular responses inside the cell. And not in this it's not stated in this picture, but mostly what these proteins do is they phosphorylate things. And so they just put on a phosphate or take off a phosphate on any kind of molecule, and then that phosphate somehow through biochemistry causes the molecule to be activated and to produce a response. So that is the G protein coupled receptor with the G protein phospholipase C pathway, and that is for the alpha 1 receptors that become activated, and you get diacylglycerol, DAG, and inositol triphosphate concentrations that increase, which are second messengers. So now let's go to adenylyl cyclase pathway. So a protein comes in, or a norepinephrine or epinephrine or dopamine in this case come in. They bind to this receptor, which is a G protein coupled receptor, and there's usually 7 passes between the membrane. And this is what I was talking about in that last cartoon that it didn't show, but there's an alpha, beta, and gamma subunit, and the alpha subunit, it is GDPs attached to it, and so when it becomes activated, the GDP gets kicked off, the GTP gets kicked on, and this alpha subunit separates from this gamma and beta part of this G protein. And then in this case, in the adenylyl cyclase and not the phospholipase pathway, in this adenylyl cyclase pathway, the GTP then turns on adenylyl cyclase. All this is inhibitory. We'll talk about that in a minute. But the GTP turns on adenylyl cyclase, and what the adenylyl cyclase does is it takes ATP and converts it into cyclic AMP, CAMP. And what happens is 2 phosphates come off, and then what CAMP does, or cyclic AMP does, is protein kinase A, also in the last pathway, in the phospholipase C pathway, it's sitting around in the cytoplasm, and it becomes active through cyclic AMP. And what cyclic AMP does, or what protein kinase A does, the active form, is it takes a protein with an OH group, and it converts that OH group into a phosphate group. It switches them out, and then that protein becomes active, and you get a cellular response. Phosphodiesterases, they take cyclic AMP and convert it into 5' AMP. So that's kind of the pathway of adenylyl cyclase and the kind of this G protein pathway. And then you can have an inhibitory molecule that comes in here, binds to that, activates the inhibitory receptor. This kind of comes over and it turns on, it inhibits this adenylyl cyclase. So these are kind of the classic G protein pathways and how that relates to this norepinephrine, epinephrine, sympathetic postganglionic synapse. And the beta receptors type of these adrenergic receptors, they stimulate the adenylyl cyclase pathway that we just talked about, and the alpha 2, if an alpha 2 receptor or that inhibitory receptor on that last cartoon gets activated, then it inhibits adenylyl cyclase. So let's talk a little bit about the sympathetic and parasympathetic divisions, and we'll talk more specifically about each one, but this picture kind of gives us a good overview. And what we talked about in the last few slides about the receptors and the transmitters and all that and how they're kind of all there, let's kind of zoom out and we'll see where all of these sympathetic and parasympathetic division runs to. So the sympathetic division, like I said, we have to run sympathetic innervation to the skin, the musculoskeletal system. We have to run it to all over the body. Whereas the parasympathetic division, we just have to run to several parts, several organs in our body. So first we'll go over kind of the anatomy of the sympathetic division. And so through the spinal, starting in the spinal cord segment of T1, the T1 spinal nerve, you have sympathetic neurons exiting those areas. And so it goes clear down to the lumbar section of your spine. Remember, we have to have two nerves, two neurons. We have pre and post. So the pre-neuron, it comes out and it can either go, it comes out into the spinal, comes out of the spinal cord and it goes up to 3 ganglion. So it comes out of the, starting at T1, the preganglionic cell of the sympathetic division comes out and it can kind of either synapse on these 3 locations. And we'll talk about these 3 locations later. But then they synapse here, and remember that this, in the sympathetic nervous system, these are, the neurotransmitter is acetylcholine and nicotinic. So right here, these are all acetylcholine is the neurotransmitter and nicotinic, the nicotinic receptor. And then now you run into the postganglionic. So this dotted line is postganglionic and the solid line is the preganglionic. So the postganglionic cell innervates the eye, the lacrimal gland, the submandibular and sublingual glands, and the parotid gland. So you can kind of see that there. And then the cells come out, and then all the way down through the spine. They can either synapse here in a structure or a chain, as paravertebral sympathetic trunk, is they can synapse adjacent to the spine. So the preganglionic can kind of either come into here, travel up, or travel down, or synapse right there at the, let's say this is like T12 spinal nerve, and come out, and it can synapse right there. Okay, and so there's a bunch of nerves that make up what they call the greater splanchnic nerve. These are all preganglionic sympathetic neurons, and they make up this nerve here, and they synapse in what they call the celiac ganglion. Number 1 is the celiac ganglion. And then they all synapse. This is a nicotinic acetylcholine, remember. And they all synapse here, and then the postganglionic go out to the gallbladder, the stomach, the small intestine, and the kidney. Okay. The lesser splanchnic nerve is a collection of preganglionic sympathetic neurons that come out here. Some go right to the adrenal gland, and then they synapse on the medulla and release norepinephrine and epinephrine. But some of them come down here and innervate to the superior mesenteric ganglion. And then the postganglionic, remember right here too that this is a nicotinic acetylcholine because it's a preganglionic synapse with the postganglionic nerve or neuron. And so it comes down and innervates the large intestine. And then the bottom of the sympathetic innervation is you get these nerves coming out. They're preganglionics. They synapse here at the inferior mesenteric ganglion, and then the postganglionic innervate the bladder, the genitalia, and so that is all preganglionic. And that is the inferior mesenteric ganglion right here. And these are all acetylcholine nicotinic. And on this same side, these nerves, this sympathetic division are in the middle of your spine. They synapse right here on either side of the sympathetic paravertebral sympathetic trunk, and then all these postganglionics go out to the skin and to the musculoskeletal system. So now let's talk about the parasympathetic division. Now the parasympathetic division is also known as the craniosacral part of the autonomic nervous system. Cranio because cranial nerve number 3, number 7, number 9, and number 10, they all have parasympathetic divisions or parasympathetic fibers running in these cranial nerves. Now sacral is because S2, 3, and 4, S2 through S4 of the sacral nerves also have parasympathetic innervation or fibers running in the nerve. And remember that these are all preganglionic. So the solid lines are preganglionic, and when they synapse, remember it's nicotinic and acetylcholine because all presynaptic to postsynaptic autonomic nervous systems, acetylcholine is the neurotransmitter and nicotinic is the receptor. You have the ciliary ganglion, the pterygopalatine ganglion, the submandibular ganglion, and the otic ganglion. And in the vagus nerve, cranial nerve number 10, cranial nerve number 10 does a lot of work, and it comes out here and parasympathetically innervates, all of these visceral organs. And then S2, S3, S4 nerves, they come out here and innervate the large intestine, the bladder, and the genitalia. So when we talk about pharmacology and one drug, let's say, blocks nicotinic receptors, everywhere that you see a preganglionic synapting with a postganglionic, it's going to affect that. And so there's tons of side effects with these autonomic nervous system drugs because it's kind of all interconnected. And so there's not a lot of specificity. So if you need heart rate to go up, let's say you got a beta blocker for hypertension. Well, the beta blocker, it blocks the beta adrenergic receptors here on the heart, let's say, or on the smooth muscles, but there's adrenergic receptors everywhere you see a post-sympathetic innervation on a gland or an organ. You have adrenergic receptors, and so there's a lot of side effects with autonomic drugs. But you gotta cost-benefit analysis. You gotta if the patient doesn't get the blood pressure down, he could have cardiovascular problems or whatever. So you got to weigh the pros and the cons of each thing. But so here's a good picture about the sympathetic division and the parasympathetic anatomy, and try to match up the receptor and the neurotransmitter at each one of these junctions. Okay, we'll see you in the next video.